TALE-likes. Transcription Activator-Like Effector-Likes TALE-likes are a group of bacterial DNA-binding proteins named for the first and still best studied group, the TALEs of Xanthomonas bacteria. TALEs are important factors in the plant diseases caused by Xanthomonas bacteria, but are known primarily for their role in biotechnology as programmable DNA-binding proteins, particularly in the context of tail nucleases. TALE-likes have additionally been found in many strains of the Ralstonia solanacearum bacterial species complex in Paraburkholderia rhizoxinica strain HKI 454 and in 2 unknown marine bacteria. Whether or not all these proteins form a single phylogenetic grouping is as yet unclear. The unifying feature of the TALE-likes are their tandem, arrays of DNA-binding repeats. These repeats are, with few exceptions, 33-35 amino acids in length and composed of 2 alpha-helices on either side of a flexible loop containing the DNA-based binding residues and with neighbouring repeats joined by flexible linker loops. Evidence for this common structure comes in part from solved crystal structures of TALEs and the Burkholderia TALE-like BAT, but also from the conservation of the code that all TALE-likes used to recognize DNA sequences. In fact, TALE, RipTAL, and BAT repeats can be mixed and matched to generate functional DNA-binding proteins with varying affinity. TALEs. TALEs are the first identified, best studied, and largest group within the TALE-likes. TALEs are found throughout the bacterial genus Xanthomonas, comprising mostly plant pathogens. Those TALEs which have been studied have all been shown to be secreted as part of the type Roman 3 secretion system into host plant cells. Once inside the host cell, they translocate to the nucleus, bind specific DNA sequences within host promoters, and turn on downstream genes. Every part of this process is thought to be conserved across all TALEs. The single meaningful difference between individual TALEs, based on current understanding, is the specific DNA sequence that each TALE binds. TALEs from even closely related strains differ in the composition of repeats that make up their DNA binding domain. Repeat composition determines DNA binding preference. In particular, position 13 of each repeat confers the DNA-based preference of each repeat. During early research, it was noted that almost all the differences between repeats of a single TALE repeat array are found in positions 12 and 13, and this finding led to the hypothesis that these residues determine base preference. In fact, repeat positions 12 and 13, referred to jointly as the repeat variable residue RVD, are commonly said to confer base specificity despite clear evidence that position 13 is the base determining residue. In addition to the repeat domain, TALEs also possess a number of conserved features in the domains flanking the repeats. These include domains for type Roman 3 secretion, nuclear localization, and transcriptional activation. This allows TALEs to carry out their biological role as effector proteins secreted into host plant cells to activate expression of specific host genes. Diversity and evolution. Whilst the RVD positions are commonly the only variable positions within a single TALE repeat array, there are more differences when comparing repeat arrays of different TALEs. The diversity of TALEs across the Xanthomonas genus is considerable, but a particularly striking finding is that the evolutionary history one arrives at by comparing repeat compositions differs from the found when comparing non-repeat sequences. Repeat arrays of TALEs are thought to evolve rapidly, with a number of recombinatorial processes suggested to shape repeat array evolution. Recombination of TALE repeat arrays has been demonstrated in a forced selection experiment. This evolutionary dynamism is thought to be made possible by the very high sequence identity of TALE repeats, which is a unique feature of TALEs as opposed to other TALE-likes. T0. Another unique feature of TALEs is a set of 4 repeat structures at the N-terminal flank of the core repeat array. These structures, termed non-canonical or degenerate repeats, have been shown to be vital for DNA binding, though all but one do not contact DNA bases and thus make no contribution to sequence preference. The one exception is repeat -1, which encodes a fixed T0 preference to all TALEs. This means that the target sequences of TALEs are always preceded by a thymine base. This is thought to be common to all TALEs, with the possible exception of TAL-C from Xanthomonas ORZ-PV. ORZ strain EX on 1947. Discovery and molecular properties. It was noted in the 2002 publication of the genome of reference strain Ralstonia solanacearum GM-1000 that its genome encodes a protein similar to Xanthomonas tails. Based on similar domain structure and repeat sequences, it was presumed that this gene and homologs in other Ralstonia strains would encode proteins with the same molecular properties as tails, including sequence-specific DNA binding. In 2013, this was confirmed by 2 studies. These genes and the proteins they encode are referred to as RipTAL Ralstonia, the injected protein tail-like, in line with the standard nomenclature of Ralstonia effectors. Whilst the DNA binding code of the core repeats is conserved with tails, RipTAL do not share the T0 preference. Instead, they have a strict G0 requirement. In addition, repeats within a single RipTAL repeat array have multiple sequence differences beyond the RVD positions, unlike the near-identical repeats of tails. RipTAL have been found in all 4 phylotypes of R, solanacearum, making it an ancestral feature of this clade. Despite differences in the flanking domains, the sequences their RVD target are highly similar. Biological role. Several lines of evidence support the idea that RipTAL function as effector proteins, promoting bacterial growth or disease by manipulating the expression of plant genes. They are secreted into plant cells by the type Roman 3 secretion system, which is the main delivery system for effector proteins. They localize to the cell nucleus and are able to function as sequence-specific transcription factors in plant cells. In addition, a strain lacking its RipTAL was shown to grow slower inside eggplant leaf tissue than the wild type. Furthermore, a study based on DNA polymorphisms in RipTAL repeat domain sequences and host plants found a statistically significant connection between host plant and repeat domain variants. This is expected if the RipTAL of different strains are adapted to target genes in specific host plants. Despite this, no target genes have been identified for any RipTAL. Discovery. The publication of the genome of bacterial strain Paraburkholderia rhizoxinica HKI 454 in 2011 led to the discovery of a set of tail-like genes that differed considerably in nature from the tails and RipTALs. The proteins encoded by these genes were studied for their DNA binding properties by 2 groups independently and named the Burkholderia TALE-likes, or BAR-H. This research showed that the repeat units of the Burkholderia TALE-likes bind DNA with the same code as tails, governed by position 13 of each repeat. There are, however, a number of differences. Biological role. Burkholderia TALE-likes are composed almost entirely of repeats, lacking the large non-repetitive domains found flanking the repeats in tails and RipTALs. Those domains are key to the functions of tails and RipTAL, allowing them to infiltrate the plant nucleus and turn on gene expression. It is therefore currently unclear what the biological roles of Burkholderia TALE-likes are. What is clear is that they do not affect the proteins secreted into plant cells to act as transcription factors, the biological role of tails and RipTALs. It is not unexpected that they may differ in biological roles from tails and RipTAL, since the lifestyle of the bacterium they derive from is very unlike that of tail and RipTAL-bearing bacteria. The rhizoxinica is an endosymbiont, living inside a fungus and like Rhizopus microsporus, a plant pathogen. The same fungus is also an opportunistic human pathogen in immunocompromised patients, but whereas B. rhizoxinica is necessary for pathogenicity on plant hosts, it is irrelevant to human infection. It is unclear whether the Burkholderia TALE-likes are ever secreted either into the fungus, let alone into host plants. Uses in biotechnology. As noted in the publications on Burkholderia TALE-likes, there may be some advantages to using these proteins as a scaffold for programmable DNA-binding proteins to function as transcription factors or designer nucleases compared to tails. It has been fused with a FOC, a nuclease analogous to TALEN. Advantages include a shorter repeat size, more compact domain structure, no large non-repeat domains, greater repeat sequence diversity, enabling the use of PCR on the genes encoding them and making them less vulnerable to recombinatorial repeat loss. In addition, Burkholderia TALE-likes have no T0 requirement, relaxing the constraints on DNA target selection. However, few uses of Burkholderia TALE-likes as programmable DNA binding proteins have been published outside of the original characterization publications. Discovery. In 2007, the results of a metagenomic sweep of the world's oceans by the J. Craig Venter Institute were made publicly available. The paper in 2014 on Burkholderia TALE-likes was also the first to report that two entries from the database resembled TALE-likes, based on sequence similarity. These were further characterized and assessed for their DNA binding potential in 2015. The repeat units encoded by these sequences were found to mediate DNA binding with base preference matching the tail code and judged likely to form structures nearly identical to baton repeats based on molecular dynamic simulations. The proteins encoded by these DNA sequences were therefore designated marine organism TALE-likes M or TL1 and 2 GenBank. Similar sequences found in metagenomes included evolutionary relationship to other TALE-likes. Whilst repeats of M or T alone and 2 both conform structurally and functionally to the TALE-like norm, they differ considerably at the sequence level both from all other TALE-likes and from one another. It is not known whether they are truly homologous to the other TALE-likes and thus constitute, together with the tails RipTAL and BATs, a true protein family. Alternatively, they may have evolved independently. It is particularly difficult to judge their relationship to the other TALE-likes because almost nothing is known of the organisms that M or T alone and M or T elsewhere come from. It is known only that they were found in 2 separate seawater samples from the Gulf of Mexico and are likely to be bacteria based on size exclusion before DNA sequencing. Legal status. A patent for BAT and marine TALE-likes in protein engineering was filed in July 2012. It is currently pending in all jurisdictions.